Hello, my friends. Alex Javankov here, and welcome to Insilico. Today we're doing something new. We're experimenting as always, and we're testing the multi-platform broadcasting system. If it works, I'll be doing these from time to time whenever we have something cool to announce. And I'll be doing that from multiple offices of Insilico around the world. But today we're doing an experiment, so don't judge. If you cannot see something clearly, we'll improve. And I think that we will do those more often. So let me bring you closer to the screen and ensure that you can see the monitor. Let's see if you can see the screen here. I'm going to be broadcasting on YouTube, Twitter, and my Insilico, my LinkedIn profile. And just to remind you what Insilico is, we are a truly global company with multiple original sites. Our headquarters is in Boston and Cambridge, so we hope to be able to do a welcoming reception sometime this fall. We have pretty good financials, going from $4.7 million in revenue in 2021 to $30 million and $51 million 2023. Multiple deals signed. We're truly global, so we currently have sites in New York and Montreal. That's where our theoretical AI is being done. We have a very substantial site in the Middle East. We're all over the Middle East. We have more than 70 scientists there now. Our first APAC site was in Taipei, and then we scaled. We're now in Hong Kong, Shanghai, Suzhou. We have only around 10 people in Hong Kong, but they're doing very valuable work in target discovery, and we're very proud of our values. Just to remind you what we do, we sell software. And we produce valuable therapeutics via our pipeline, and many of them are available for licensing. So on the software side, we have biology, chemistry, and medicine, so pretty much covering the entire drug discovery and development space with AI. We also now have Science 42 with really, really cool tools like Dora. Dora is a multi-agent generative research assistant that allows you to write research papers from scratch. And we just open sourced our precious 3GPT, which is, in my opinion, the first live model. So check it out on Hugging Face. And we utilize the software to very rapidly design and develop valuable therapeutics of high or moderate level novelty, because big pharmaceutical companies, they don't like the target to be too novel before it reaches phase 2 complete if they want to acquire the asset. And you have to sometimes sacrifice novelty to ensure that commercial tractability is there. So our lead asset, TNIC, is now in phase 2 in both US and in China. And we have multiple assets that have reached clinical stage. What I'm going to be talking to you today about is our TEAD program. So this is the target that I'm also personally very passionate about for many reasons. I'm going to talk about why. And today, big news, it received IND clearance from the FDA in the US to start human clinical trials. So that means that now we have 9 drugs out of generative AI, either with a novel target or with a target that is moderately novel, going to human clinical trials, and which is super exciting. There are very, very few companies that can sport even one, and we now have 9. As a matter of fact, since 2021, we nominated 19 preclinical candidates, 9 programs are in human clinical trials. But today we're focusing on TEAD. And our program is a pan-TEAD inhibitor. It's a non-covalent binder to multiple TEAD proteins. And that is super exciting. So for many reasons. Well, first of all, I was introduced to YAP-TAZ pathway, YAP-TEAD pathway, to HIPPO pathway through aging research. it's a very important pathway modulating multiple biological processes, including the extracellular matrix. So something that I consider to be a very important hallmark of aging, because extracellular matrix during aging also gets clotted, gets dysregulated, and it's great to have molecules that can control this process. And it's also a very, very important pathway in cancer, because TEAD YAP-TAZ forms oncogenic complexes. Many cancers where Hippo pathway is dysregulated are very sensitive to TEAD inhibition. Previously, people were trying to go after TEAD1, TEAD2, TEAD3, TEAD4. So there are 4 subtypes. It looks like if you target all of them, you are likely to have much higher cancer sensitivity to the inhibitor. Also, TEAD activation promotes drug resistance in chemotherapy for multiple types of targeted and immunotherapies. So it's also a very good combination companion. So highly recommend looking at this target from the cancer standpoint, also from fibrosis, and as they say in the Middle East, inshallah, hopefully it's going to work on aging at some point in time. What is really cool is that this drug that we are developing, it's again non-covalent pan-TEAD inhibitor. So far in preclinical experiments, we've demonstrated really high safety profile, very tolerated kidney tox. So some of the competing molecules, they demonstrate some kidney tox on target and on molecule. So I think we currently will be best in class, at least in that area, and then many others. It's a favorable drug likeness and PK profile, and it's oral, very convenient dosing, and of course, since it's a small molecule, it's low cost. So it can work as monotherapy in multiple diseases. So there is established body of evidence that it works in mesothelioma, which is actually reasonably rare, but expanding therapeutic indication. It also demonstrated activity in multiple forms of head and neck cancer, endometrial carcinoma, and lung cancer, and cervical cancer. So if you know how to pick the patients right, you probably will see substantial clinical successes as monotherapy. But in my opinion, the nowadays, most of the targeted cancer therapies are combination plays, and the future is about combinations. So combination therapy potential for TEAD is immense. You can go for multiple types of lung cancer, multiple types of head and neck and colorectal cancers in combination. So substantial combination potential with EGFR and KRAS inhibitors, with PI3K alpha and BRAF inhibitors. So the combination potential is vast. What I started doing recently, we started doing kind of target practice exercises where we try to explain the target in just one slide. 1 page. So trying to explain the mechanism behind the target, the function, pathways, knockout types, therapeutic MOA, therapeutic rationale, most advanced programs by the different companies. So we also want to ensure that the field is not too crowded. And there are companies that are leading a little bit for moderately novel targets, because very often if you don't have clinical POC, nobody's even gonna talk to you. And for many of our programs, we really want to partner with pharmaceutical and biotechnology companies to ensure that those companies that specifically focus in specific therapeutic indications can benefit from our drugs. And we try to make them of higher standard of quality than big pharma usually got used to. And here we mapped out the TEAD target utilizing many parameters. What I am extremely excited about myself personally, as you know, I am very committed to aging research. I actually like to refer to aging as biology in time. So biology doesn't happen as a static process. It happens from cradle to the grave, and actually before cradle, and even a little bit before the grave. So it's very important to understand where does the target play in the hallmarks of aging and different biological processes that are driving aging and disease. So TEAD scored on pretty much every hallmarks of aging, so especially in extracellular matrix. So we decided to focus on this target instead of many others. We have hundreds of thousands of targets to go after. So it was prioritized by AI. As a matter of fact, if you are attending the ARDD 2024 in Copenhagen, which is transpiring in just over 2 weeks, so 26th of August to 30th of August, many big pharmaceutical companies are going to be there. So if you're attending that, what I'm going to be presenting is how to utilize modern methylation aging clocks that have been published by Stephen Horvath, Vadim Kladyshev, and others for mammalian tissues, for mammalian species in multiple tissues. Some of them can be now utilized for target picking. It's kind of paradoxical to me, but some of the methylation clocks are actually valuable for that. And TEAD is one of the targets that is scoring very high in those clocks, which is a surprise. So please come to my lecture at IRDD and have a look. We're going to be looking at many other targets. And currently within this space, there are several front runners. One of the front runners is Vivace, then Novartis and Beta. So all in phase 1. So hopefully there will be positive clinical readouts in the near future, and we will see much more activity around the space. So now we have received the IND approval, and we are also running in this. We believe that we have the truly best-in-class molecule. There are many reasons why you should be excited about this particular target. One is that, again, there is a very promising clinical proof of concept by Vivace. So kudos to the company which managed to do a lot of biology in this area and also started running clinical trials. So it's always good to have a front runner with chemistry that we can benchmark against. Also, Novartis is front running in this area. So this target is moderately novel. And our compound demonstrated comparable or superior efficacy in multiple preclinical tumor models. It's very different from Vivace compound. And we predicted that the clearance in humans will be significantly better, and we're going to have less tox. So that's kind of the prediction that we have right now. We also demonstrated very strong combination potential in KRAS, EGFR, and PI3K inhibitor-resistant cancers. So, and we did it in multiple preclinical models, demonstrating that this molecule has very substantial combination potential. Again, I'm a big believer in combinations. There are multiple predictive biomarkers, so now we know how to pick patients a little bit better. So 7 genes with common gene signatures, NF2, FET1, are identified. So if you are targeting mesothelioma, there is a clear biomarker. We have a range of our own biomarkers that would be available under CDA to a partner. And again, a very substantial combination potential. I think that any big pharmaceutical company targeting cancer should have it in their portfolio. And once clinical POC is achieved by more players in the field, and once there is more target confidence, I hope that the level of enthusiasm around this target will peak. I am also a believer that this target should work in multiple forms of fibrosis and fibrotic-driven diseases. So it's all about the dose and combination plays. So I think that this target has massive indication expansion potential. As a matter of fact, most of the targets that Insilico is going after have massive combination expansion potential. And for those of you who are interested in this target in this program today, again, it's a pan-TEAD non-covalent inhibitor, which is potentially best in class. Many of you who know Insilico quality and have seen several of our licensing deals with expert drug hunters who actually do head-to-head comparisons themselves. If you are interested in that, please contact Michelle Chen, our CBO, michelle@insilico.com, very easy. Keith Mickley in Boston, Keith@insilico.com, Sean Cullen, Sean at Insilico.com, or if you are too lazy to write it down right now, or just don't have the time, BD at Insilico.com. Get under CDA, look at the data room, let's start talking while the clinical trial is progressing. It's always good to have an established relationship and understanding of the molecule in the early days. And once there is more confidence in the target and people start bidding, you can submit the bid with the most intelligent approach, where you would know much more about the target, much more about the molecule, much more about the preclinical data package. It's amazing. And let's partner and collaborate. Also, please do look at our software, because I'm a big fan. I use it myself. I basically developed the software for the use by myself first, and then we industrialize it and get industry level validation. And programs like TEAD, where you have potentially a best-in-class molecule developed in a very short period of time, with very high confidence and some front runners in a non-so-crowded space. It's a good testament that the software works. Let's collaborate, and let's make people live longer and healthier lives. Good day to you.